For decades, genetic engineering has relied on a specific class of protein-based tools known as type Is restriction endonucleases, such as the BSAW-I enzyme, to cut and assemble long-chain DNA. These biological tools operate under strict constraints. Restriction enzymes will only recognize and cut at highly specific palindromic sequences. When they make a cut, they typically leave behind short, 4-base single-stranded overhangs, commonly referred to as sticky ends. This diagram shows a standard Golden Gate assembly using a BSAW-I enzyme. Notice how it leaves an overhang of just 4 loose bases. In physical chemistry terms, a 4-base sequence possesses a very low melting temperature. This results in highly transient hybridization kinetics. The bonds are weak and often fail to hold the fragments together long enough for DNA ligase to seal the joint. But if we use a sequence-independent chemical cleavage method, we can generate custom overhangs up to 18 bases in length, creating a stable, interlocking bond. Bypassing biological restriction enzymes entirely removes palindromic sequence constraints and enables thermodynamically stable DNA assembly. A research team from Japan's Nagoya and Gifu universities has developed a mechanism to achieve exactly that. Their approach utilizes structured, polymer-coated silver nanoparticles. By substituting biological restriction enzymes with these engineered nanoparticles, the researchers achieved sequence-independent DNA cleavage and a 5-fold increase in ligation efficiency. To understand how this system operates, we have to examine the physical chemistry. We must investigate how the target cleavage site is synthetically built into the DNA, the physical mechanism of nanoparticle chemisorption, and the molecular surface shielding required to prevent the system from degrading under thermal stress. Manipulating the atomic surface properties of these nanoparticles drives the optimization of macro-scale genetic assembly. To cut a DNA sequence without relying on an enzyme, we must introduce a targeted chemical vulnerability directly into the DNA backbone using synthetic primers. Looking at this 2D diagram of a standard phosphodiester DNA backbone, we focus on the bridging oxygen atom at the 3' position. Through multi-step synthesis, that oxygen is replaced by a sulfur atom, creating a 3' phosphothiolate linkage, denoted as TS. This single atomic substitution creates a precise biophysical vulnerability. If we can chemically activate that sulfur atom, the adjacent phosphorus-sulfur bond becomes highly susceptible to a localized nucleophilic attack by surrounding water molecules, which will cleanly sever the strand. Because this TS linkage can be synthesized into any DNA primer, it provides a customizable chemical target that completely removes the reliance on rigid palindromic recognition sequences. In the early 1990s, researchers successfully executed this exact cleavage. They discovered that free silver ions in solution would activate the sulfur atom and cleave the TS linkage at ambient temperatures. Free silver ions bind nonspecifically, causing DNA to precipitate with just a 14.4% recovery rate. The team replaced them with structured 10 to 20 nanometer silver nanoparticles. Their metallic surface selectively coordinates with the sulfur atom, driving site-specific hydrolysis. Using solid-phase nanoparticles ensures the DNA receives the necessary Lewis acid activation to sever the strand, without flooding the solution with toxic, precipitating free ions. Achieving a clean chemical cut solves the first problem. The secondary bottleneck is isolating the cut target DNA from the unwanted waste material. Immediately after hydrolysis occurs, the target 5' phosphorylated DNA fragment floats freely in the aqueous solution, alongside the unwanted 3' thiol DNA waste fragment. The waste fragment does not stay free for long. It rapidly undergoes chemisorption, binding tightly to the surface of the nanoparticle. The physical chemistry driving this is highly specific. The newly cleaved waste fragment contains a reactive thiol group, which forms a strong, covalent silver-sulfur bond with the metallic surface, nanometers. Its zeta potential drops from negative 31.72 down to negative 51.05 millivolts. confirming the nanoparticle is heavily coated in highly negative DNA backbones. This creates an elegant physical extraction method. Because the waste fragments are covalently anchored to heavy, solid nanoparticles, running the sample through a centrifuge easily pellets the waste complex to the bottom. The pure target DNA remains in the clear supernatant liquid above it. This built-in self-purification mechanism takes the dismal 14.4% recovery rate of the historical free ion method and drives it to a near-perfect 98.4%. The system still faced one final physical chemistry bottleneck. Bare silver nanoparticles possess extremely high surface energy. When exposed to the high ionic strength of standard PCR buffer salts, they become unstable and aggregate. Researchers initially heated the reaction to 95 degrees Celsius for 2 hours for maximum cleavage efficiency. However, this severe thermal input risks denaturing long-chain DNA. They resolved this by coating the nanoparticles in a water-soluble polymer known as polyethylene glycol, or PEG. These neutral PEG molecules provide critical charge shielding, resisting salt-induced aggregation. With the nanoparticles stabilized, thermal requirements drop significantly. The pegulated system achieves over 91% cleavage efficiency at a safe 50 degrees Celsius, preserving the long-chain DNA's structural integrity. By utilizing PEG shielding, the research team successfully translated an aggressive physical chemistry reaction into a practical tool for biological application. Moving from chemical synthesis to functional genetic output, the true value of the T linkage is that it can be positioned in any arbitrary sequence. This allows researchers to execute scar-free DNA assembly. The thermodynamic payoff is clear on this comparison chart. A standard 4-base enzymatic overhang yields a joining efficiency of just 8% when treated with T4 DNA ligase. But an 18-base custom overhang, generated by the silver nanoparticles, increases that efficiency to 44%. To validate these metrics in vitro, the team used the pegulated nanoparticle method to assemble an 848 base pair DNA cassette encoding green fluorescent protein. The researchers then transfected that assembled cassette into human HeLa cells. As this fluorescent microscopy image shows, the cells successfully expressed the green fluorescent protein, confirming the accuracy of the sequence-independent assembly. A 5-fold increase in ligation efficiency provides the thermodynamic stability required to move from splicing small fragments to constructing accurate, genome-scale DNA that functions in live cells. The next engineering challenge for this platform is scaling. To synthesize entire artificial genomes, the process must be optimized to handle simultaneous, multi-fragment ligations in a single pot reaction. Trace silver nanoparticles can inhibit downstream enzymes or induce cytotoxicity during mammalian transfections, making absolute clearance through rigorous extraction protocols a biological necessity. Overcoming these extraction bottlenecks will remove the constraints of rigid enzymatic splicing, offering researchers a sequence-independent, thermodynamically optimized method for assembling the genetic blueprint.
Scientists are racing to unlock technologies. Breakthroughs in AI are reshaping our future. The future is arriving faster than expected.